Bayer AG's Animal Health Research Center in Monheim, one of the largest and most modern private institutes of its kind in the world. Here, scientists are working to develop products against many animal diseases that occur worldwide. They have three aims: to protect animals from disease, to develop curative medicines, and to produce drugs to control transmission vectors. One of the diseases that can threaten herds is foot and mouth disease. The business group Animal Health produces vaccines to control foot and mouth disease in specially licensed and regularly inspected plants. The antigens for these vaccines are stored, ready to provide rapid and effective prophylaxis in case of outbreaks of this viral infection anywhere in the world. FMD is a nightmare for all farmers, veterinarians and government veterinary officers, because this disease has disastrous effects on the livestock population of every affected country. In some countries, this may even result in the stamping out of complete herds. The causative agents are viruses, a discovery made at the end of the last century by German bacteriologist Friedrich Loeffler, a colleague of Robert Koch. During the research phase of this disease, guinea pigs became of such importance that a guinea pig memorial was even built at Reims. The production and supply of FMD vaccine by Bayer is thus rooted in a great tradition of microbiological research in Germany. In 50 countries, Bayer experts are today in constant contact with farmers, veterinarians and government veterinary officers responsible for maintaining the health of livestock herds. When foot and mouth disease occurs, the effects are felt throughout the national economy. There's no drug to cure this disease. Priority has to be given to prophylaxis, to preventing an outbreak of the disease in the first place. Let's start by looking at the symptoms of FMD. Foot and mouth disease is a disease of all cloven-footed animals: cattle, sheep, goats, and pigs. Wild animals provide a reservoir of the virus. Transmission of FMD occurs through animate or inanimate vectors, directly from animal to animal or from man and his implements to animals. Markets and shows are places where the virus may be easily transmitted from sick animals to healthy ones. There is also a risk attached to purchasing animals of unknown origin. The first signs of infection are lesions called primary aphthy at the port of entry. They often go undetected at first. There's no fever at this stage. The virus is distributed throughout the body in the blood. The incubation period is 3 to 5 days. At this stage, the animals appear tired and apathetic. Fever develops, and the body temperature may rise to 41 degrees Celsius. There's a discharge of long threads of mucus from the mouth, and tough mucus often appears in the nares as well. Secondary aphthy are a typical sign that specific organs are affected. The picture shows incipient vesicle formation on the muzzle and the mucous membranes of the lips. The tongue is most severely affected. Vesicles also form around the claws, especially in the interdigital space and around the coronet. Blisters also appear occasionally on the skin of the teats and the mucous membrane of the rumen. The aphthy are filled with fluid, which is clear at first, becoming cloudy later. They soon burst, leaving weeping, very tender and sore patches. If several feet are affected simultaneously, the pain can be so excruciating that the animals remain recumbent and are very reluctant to get up. Let's look at the sort of quarantine measures that are needed. Details may vary from one country to another. Any suspected case of FMD must be immediately reported to the local veterinary officer. He will order all the immediate measures necessary to prevent the disease from spreading. A prohibited area is set up to isolate the pathogen-bearing herd. The premises are declared infected. This has numerous consequences, some of them unpleasant. The entrance to the farm is disinfected to prevent the spread of the disease by vehicles. All traffic leaving the farm is severely restricted. Trade in animals or materials is forbidden. The animal houses, too, must be treated with disinfectants at least once daily. An extremely high level of cleanliness is required. All the cloven-footed animals on the farm have to be slaughtered. This experience is a terrible shock for the farmer. Let's look at how the veterinarian reaches his diagnosis. He takes samples and sends them to the diagnostic laboratory. The tongue of this animal already shows the typical vesicles. The samples are sent to the laboratory as quickly as possible, since a suitable vaccine cannot be produced until the virus type and subtype have been identified. The most important thing is to slaughter or vaccinate all the healthy animals near the infected farm to limit the spread of the disease. The laboratory identifies the virus type and subtype as rapidly as possible. There are a multitude of types, subtypes and strains of the FMD virus worldwide. The vaccine used must contain inactivated virus of exactly the type and subtype that has been diagnosed in each specific outbreak. The following approaches for controlling FMD are used: stamping out and ring vaccination, preventive mass vaccination. As we said, all cloven-footed animals on an infected site must be destroyed immediately. Depending on the control strategy and extent of infection, compulsory slaughter is carried out over a wide area in some countries. Small-scale measures are never sufficient, as the virus can easily be carried over large distances. These dramatic pictures show the horrendous economic damage that an outbreak of FMD can cause. The second means of controlling FMD is stamping out combined with ring vaccination. All animals at risk are vaccinated, starting with herds that are not yet affected. Vaccination is carried out systematically in a spiral around the infected farm, working from the outside in. The wind direction is important, as pathogens may be blown across country. The third strategy for combating FMD is preventive mass vaccination. Regular prophylactic immunization of the entire cloven-footed animal population has proved effective in preventing outbreaks of FMD entirely in high-risk countries, or countries where the disease is endemic. For this reason, Bayer holds permanent stocks of vaccines for all relevant virus types in nitrogen. It means that the right vaccine can be supplied to the local veterinarian as soon as the virus type has been identified. The Bayer research team carries out intensive research into viruses. A large supply of inactivated, frozen FMD viruses and FMD vaccines are kept in stock and represent an instantly available safety reserve that can be called on at any time. The vaccines are delivered from Cologne to many parts of the world. The viruses are propagated in modern closed systems on susceptible cell suspensions. During this process, these tissue cells are destroyed. However, the vaccine must not contain debris from destroyed cells. In the past, impurities of this kind often triggered allergic protein reactions which led to complications. It's vital for the cell debris to be separated meticulously from the virus. Viruses in vaccines also have to be reliably inactivated. We found ethylenimine to be an inactivating agent which considerably improves the safety of vaccines because it inactivates them reliably. The use of this agent enables us to rule out diseases which used to occur as a result of inadequately inactivated vaccines. Bayer's scientists found polyethylene glycol to be an ideal agent concentrating and precipitating the viruses, which are then centrifuged to separate them from non-virus proteins. Concentrating viruses makes it easier to preserve them for long periods. These two stages, reliable inactivation and concentration, are milestones of Bayer's research into the manufacturing process used for FMD vaccines. The virus concentrate undergoes intermediate storage under quarantine conditions. Once it has been released by quality control, it is stored in liquid nitrogen. The entire production process complies with the latest quality and safety requirements of the European Union and is under continual surveillance by EU experts. Checks are also carried out by representatives of local government agencies. In-process controls involving laboratory diagnosis accompany all stages of production to ensure that quality is maintained. This is where purity and sterility are tested. The ELISA method to test identity, infectivity titration, and 146S determination are also carried out here. The Federal Research Institute for Viral Diseases of Animals in Tübingen. Here, the vaccine is tested on the target animal. This government test facility regularly investigates BioVac FMD vaccines for efficacy and safety in the target animal in strictly isolated animal houses. Official controls cover the sterility and safety of the final product. Safety comprises inactivation kinetics, safety in tissue cultures and in the target animals, and local and systemic tolerability. Potency is also evaluated in a PD50 test in target animals according to the EP and using the validated correlated SN90% in vitro test. Back to Bayer in Cologne. This is where the reserve of FMD vaccine is kept exclusively for Germany. In other words, a supply of antigen concentrate large enough for 1 million doses of monovalent vaccine for each subtype of the foot and mouth pathogen is kept here. 100,000 doses of monovalent, ready formulated vaccine are also available. Each of these subtypes forms part of the government vaccine reserve. The ready-to-use vaccines can be supplied to veterinarians anywhere in Germany within 24 hours. BioVac vaccines are approved for use with all ruminants, and they cover the 10 most important strains of FMD in the world. Because the production facility in Cologne supplies vaccines to many parts of the world in which foot and mouth disease is a problem. Facilities are also available for converting newly emerging strains of the pathogen into vaccines at short notice. The European Union is in the process of establishing a European vaccine reserve. Government facilities will be established at 3 sites in France, the United Kingdom, and Italy. Bayer's reserve in Cologne is the 4th site in this European prevention concept. 1/4 of the planned 50 million vaccine units will be stored in the form of virus concentrates in liquid nitrogen in Cologne. In the event of a disease outbreak, they will be thawed, quickly formulated under aseptic conditions, and bottled. They can, for example, be filled into 500